So I'd like to welcome everyone to the CVRN Rising Stars seminar series. We've got quite a diverse set of presentations today. As you can see, the title is From Molecular Pathways to Population Health. I'd like to start the meeting by giving an acknowledgement of country. So the Institute acknowledges the traditional owners of country throughout Australia and recognizes the continuing connection to lands, waters, and communities. We pay our respect to the Aboriginal and Torres Strait Islander cultures and to elders past and present. So to start with, just a brief thing about the CVRN network, as I'm sure most of you are aware. We're a mixture of clinical, public health, biomedical researchers. 75% of us are the early mid-career researchers, with 25% seniors. We're a large collaboration of different institutes that brings a diverse wealth of knowledge to the table. And this is our governance framework. There are opportunities throughout the years to get involved with the CVRN committee and various subcommittees to organize events such as this. So when those opportunities arise, I definitely recommend to get involved. There's a picture of some of the subcommittee there. So today we've got a range of talks, but the first talk is going to be by Dr. Albert Henry, who is currently working at the Garvan Research Institute, but was previously a research fellow at University College London. Dr. Henry's research interests lie at the intersection of data science, human genetics, and systems biology. And today, I believe he's currently leading a study to disentangle causal biology underlying complex traits by leveraging paired genetics and multi-omics data at the single cell resolution. So I'm going to pass over to Dr. Albert Henry, and he can take away with some of his findings.
Thanks for the introduction and for the kind introductions. So as previously mentioned, I'm Albert Henry, currently working at the Garvan Institute of Medical Research. And this is, I just moved last year from University College London. And this is my first CVRN seminar. So I'm very pleased to introduce some of the works that I've done. I think most of these works I've done when I was a postdoc at UCL. And just to clarify earlier in the introduction, it's not causal biology, it's a causal biology. So because we try to use genetics to dissect the causal mechanism of disease. And we use that using, oops, I think it's probably, oh, it's the wrong way around, sorry. Right, so the reasons we use human genetics to do some of this study is that we know that disease as focusing on is heart failure, and heart failure, as we know in cardiovascular research, is a very heterogeneous disease. And if we look at the symptoms in the clinics when patients present with heart failure coming to the clinic, oftentimes it's just the tip of the iceberg. It often comes with a fluid congestions, but underneath that, there's actually lots of etiologies going on. Some of this etiology, like ischemic heart disease or cardiomyocytes, are well defined, but there are also some other less clearly defined etiology of heart failure. Sometimes it's a risk factors, could be renal disorder, type 2 diabetes, or obesity, but we don't actually know what's the causal mechanisms. So we want to use genetics because we know that genetics actually affects the heart failure. It has a heritable component that is shared with all these different etiology. So in this presentation, I just present kind of like how we can use genetics to understand kind of like molecular etiology for the heart failure, and I touched a bit on cardiomyopathy as an example of a big component of heart failure. So for some of you that might not be so familiar with the concept of genome-wide association study, or GWAS, this is kind of like bread and butter in genetic association research today. So essentially, we usually want to find in the populations the genetic variations or single nucleotide polymorphisms, or SNPs, using genotyping chips that are more likely to present in the case of interest than in the healthy individuals. So for examples, in heart failure, I want to find the genetic variations or SNPs that are more likely to present in patients with heart failure compared to the general population. And this effort initially starts in about 5 years ago, 2020, led by my collaborators in the UK, Tom Lambert and Dr. Sonia Shah. Now Sonia led her groups in Brisbane. But essentially at the time, we collected a number of patients from global consortiums or including 20 studies in 2020. Now it grew to almost 40 studies, and the latest through this consortium efforts called the Heart Failure Molecular Epidemiology for Therapeutic Targets, or HERMES consortiums. We managed to collect 150,000 cases, and this is from a paper that we recently published earlier this year. We collected 150,000 heart failure cases from international cohorts, and then we stratified based on etiology in the non-ischemic heart failure, and then further stratified based on left ventricular ejection fractions. The reasons we want to focus on the non-ischemic heart failure is because we know quite a bit about the ischemic component, and that's quite clear. Usually it's caused by coronary artery disease, so we want to focus more on the non-ischemic components here. And the second types of GWAS and we split that a bit, but this is focusing on dilated cardiomyopathy, which we know is a major component of heart failure, and it has a huge heritable component as well. So we did secondary analysis using kind of like a slightly different cohort composition, but this time we collected patients with dilated cardiomyopathy, and this is done in collaboration with Zeng and James Ware lab at Imperial College London. So in our GWAS, this is from the heart failure GWAS, we identified about 66 genetic susceptibility loci. 37 of these not been previously reported in previous GWAS. And we can see that this is already like a substantial increment from the previous analysis, where we identified only about 11 genetic susceptibility loci throughout the genomes. But we can see that the SNP heritability on the proportion of variants in the phenotypes explained by the SNPs or genetic variation is actually quite low. It's about 5% for the heart failure in the general heart failure, and it got about 10% for the non-ischemic heart failure with the dissection fractions. And we can see from the Manhattan plot, this is kind of like how the SNPs or the genetic loci are distributed across the genomes. The pattern is slightly different across these different heart failure subtypes. And this is the GWAS results for the DCM. And earlier I mentioned a bit that the DCM case here is actually fewer than the heart failure. It's about 10% the number, 14,000 versus 150,000. But what we can see here, the number of loci is actually very similar. And the heritability for DCM here is higher. And it's even higher, about 20% if we use stricter definition of DCM. And we think this is because of the different genetic architectures underlying these different phenotypes. And we can see that if we look at, plot this as a function of minority frequency in the populations, we can see that this is a classical trumpet shape for that connects the rarer genetic variant. Usually it harbors a larger effect size in the population. So, but we can see if we compare the two between in the left heart failure, the right is DCM. With the DCM, we can see that some of this, if we combine to a polygenic risk score, don't know if I can zoom in. Polygenic risk score here, which is a combined of these dots here, actually have a very similar effect size of this rare variations, which we know of which mutations we know cause the DCM. So this is the two of these phenotypes are both polygenics, and for DCM there are also a strong monogenic component. And this is after that, we want to know kind of like the genes that actually cause in each of these loci. So we combine different methods here, about 6 that we combine, and in the end, we identified about 66 prioritized genes for heart failure. And this is how it was distributed across these different subtypes. one of the locus that we're quite interested in is the TMAM18, and this is quite a we think this is one of the potential novel finding because a knockout model in mice has been linked to obesity, which we know is a big risk factor for heart failure with preserved ejection fraction. And I'll jump from the heart failure and now to the DCM just to give an examples. Once we've done kind of like this different identifying the potential genes, we can then do gene set enrichment analysis. So this is an examples from the GWAS, and then we took that results to kind of like pathway enrichment analysis, and we identify the convergence of these genes that points to different functions in the across the DCM pathology. So we think these are functions that are related to intracellular signaling, cell cycle and apoptosis, ER stress, cell-cell and cell matrix interactions, and sarcomere and cytoskeletal. And the other analysis that we can do after we've done the GWAS is doing what we call a heritability enrichment analysis. So this is combining the GWAS results with another sort of like genetic associations using tissue level gene expression. And we identified a different pattern of heritability enrichment here. And then what's striking to us is that non-ischemic heart failure with preserved ejection fraction is actually enriched for kidney and pancreas. And I'll probably just skip this, skip the detail of this, but we performed this analysis to a different bunch of traits, to identify causal traits, and we use network analysis to model the associations between the traits. And our initial goal is to identify the etiologic models that underlies these different disease mechanisms, and we use network analysis to identify that actually some of these genes clustered with this non-risk factor of heart failure. So just to summarize, we performed genetic association study, and we did a bunch of analysis, including genome-wide approach, log-centric approach to identifying the underlying cause. But what we note here, we know that genetic association study is a powerful toolbox, but it also the results are really dependent on sample size, sample characteristic, and how we define the case for our phenotypes. With that, I would like to thank my collaborators, mostly from my previous institute, UCL, and some other external collaborators. And we want to take this forward. Now, this is the work that I'm sort of involved in Garvan. We want to combine the single cell genetics with the GWAS results, heart failure, and including other traits, in particular cardiovascular diseases, to identify causal mechanisms. And this is in collaboration with Joseph Paul and Gemma Victory Lab. Thank you. Question up here.
Yeah, we'll have that Q&A.
Yeah, just to clarify, we'll have all the questions at the end, and there'll be time to mingle with some canapes and drinks afterwards as well. Next speaker is Dr. Mark Schreuder, an academic researcher at the Centenary Institute and the University of Technology, Sydney. He's working in the laboratory of Professor Philip Hogg, and his expertise lies in protein biology and biochemistry, and is particularly focusing on the mechanisms of blood coagulation. So I will pass over to Mark, who will tell you more about it.
Thank you. The next one. Antiphospholipid syndrome. It's an autoimmune disorder that's characterized by the persistent presence of so-called antiphospholipid antibodies. So these are antibodies that are directed either against phospholipids or against proteins that are associated with phospholipids. And the estimated prevalence is about 50 cases in 100,000. Now, these patients are presented with a very significant increased risk of basically all kinds of different forms of thrombosis. They have heart valve abnormalities and in many cases unsuccessful pregnancies. So the disease is estimated to be responsible for about 10% of the thrombosis cases in young adults, and successful pregnancies only occur about 25%, but we can improve this to about 70% with adequate treatment. Now, the antiphospholipids antibodies that are mostly associated with cardiovascular complications include antibodies that are directed against the proteins beta-2-glycoprotein 1 and prothrombin. And in this presentation, I'll focus on the prothrombin protein. So just a very quick background into prothrombin. So prothrombin is circulating in blood as an inactive zymogen, and when coagulation is initiated, it's gets proteolytically activated, releasing some of its domains, and in the end, you get the active enzyme thrombin. And thrombin is quite a unique enzyme in a way that it has a wide range of different physiological substrates, and this makes it basically the central coagulation protein in blood clotting. Now on the right, we have a model structure of prothrombin, and as in our lab, we are very much interested in disulfide bonds. Here are in pink highlighted, the pink spheres are highlighting the disulfide bonds in prothrombin, of which there are 12 scattered around the different domains of the protein. Now, just for people who are not really familiar with the sulfide bonds, basically there are two types of covalent bonds in proteins. We're all familiar with the peptide bonds that are the bonds that link the individual amino acids to form the amino acid sequence of a protein. And there's the disulfide bonds. So this is a covalent link between two cysteine residues. And the unique thing about this disulfide bond is that it can link distant regions in amino acid sequence together. Now, the disulfide bonds, they are generally considered to be essential for stabilizing the three-dimensional structure of a protein. So traditionally, the disulfide bonds are considered as an important factor for protein folding, and as such, all potential disulfide bonds in a protein are required to be formed in order to have a mature functional protein. And in a way, that means that proteins actually exist in just identical copies of each other. And you can sort of understand that, for instance, if you take this disulfide bond number 3 out of the protein, you get like a much more flexible and different structural protein than the others. Now, because we are mostly a disulfide bond lab, we've developed this technique called differential cysteine labeling. So what we do here is we have a protein of interest, and if there are any potential unformed disulfide bonds, we label these bonds with a specific labeling agent we call carbon-12 IPA. So this labeling agent has a ring structure here composed of 6 carbon-12 atoms. And then we label the unformed disulfide bonds. Now, once we have done that, we move on to label any formed disulfide bonds. So we break the bonds using DTT, creating a new pool of free cysteines, and we label these free cysteines with the exact same labeling agents. However, in this case, this ring structure is now composed of the carbon-13 isotope. So this difference between the carbon-12 and the carbon-13 isotope generates a mass difference, and we can pick this mass difference up with mass spectrometry. Now, as I mentioned, in a traditional view, we consider all disulfide bonds to be fully formed. So if we actually would apply this technique to a protein called, like, for instance, prothrombin, we would generate a graph like this. So here we have the different disulfide bonds within prothrombin, and the y-axis shows the percentage that these particular disulfide bonds are unformed in the prothrombin molecules. Now, because we expect that all disulfide bonds are formed, we would have 0 unformed disulfide bonds, and all the data that we generate is just 0. However, when we actually do this experiment using prothrombin isolated from healthy donor plasmas, we obtain something like this. So here we can immediately see that actually all the disulfide bonds in prothrombin exist in both formed and unformed states. And for instance, what this shows here is that if we look at this disulfide bond here, it means that this particular bond is unformed in about 20% of the prothrombin molecules circulating in your blood. So essentially, to put this in a bigger picture, instead of thinking of proteins as this, in this traditional view, that are identical copies where all disulfide bonds are formed, we're actually showing that these proteins are existing in a heterogeneous mix of different structural forms that are modulated by the disulfide bonds. So for instance, you can have a protein in which only one of the disulfide bonds is formed. You can have a protein in which 3 disulfide bonds are formed, or you can have a protein that, for instance, one disulfide bond is formed, but it's another one, a different disulfide bonds. So now that we know that these proteins exist in these heterogeneous structural mixes, we hypothesized whether antibodies may preferentially bind to a subset of these structural forms. So what we did is we generated our prothrombin disulfide bond mutants, in which like individual disulfide bonds were mutated from cysteine to alanine. And we sent these prothrombin mutants to our collaborator, the group of Dr. Nicolas Pozzi in the St. Louis University, who has a small cohort of antiphospholipid patient plasma. And what they did is they isolated the antiprothrombin antibodies from these patient plasmas. They coded our prothrombin mutants to a 96-well plate. They then added the patient-derived antibodies and just see how are these patient antibodies binding to our prothrombin mutants. So this is what they observed. So here we have 6 different prothrombin disulfide bond mutants, and the y-axis shows the ratio that these antibodies are binding to the mutants compared to antibody binding to wild-type prothrombin. So here the antibody binding to wild-type prothrombin is set as 1. And we can see that 4 of the mutants display a pretty similar antibody binding compared to wild-type. But we also see that 2 of the mutants have a significantly lower antibody binding as compared to wild-type. So this shows that the antibodies are actually preferentially binding a specific subset of structural protein forms. So we wanted to look at this in a little bit more detail, and we aim to look at the open and closed conformation of prothrombin. So in blood, prothrombin circulates predominantly in a sort of closed form, with about 80% of the molecules in this sort of closed conformation. And about 20% is in a more elongated open conformation. And this is a sort of an equilibrium that constantly changes, but the percentage remain roughly the same. Now, we also know that the closed conformation is less immunogenic, just simply because it's sort of hiding its antibody epitopes. Whereas the open form is more immunogenic because it's like elongated and it sort of fully displays all the potential antibody epitopes there are. So we wanted to see whether disulfide bond mutation changes the equilibrium between the open and closed conformation. And we did that by using a technique called surface plasmon resonance, which is basically a direct protein-protein binding technique. So what we have here is we have an antibody that specifically recognizes the open form of prothrombin. We code that to the chip, and then when we flow over the prothrombin mutants, and when the mutants bind, showing it's an open form conformation, increases the signal until full binding has appeared, and we can also look at the dissociation of the prothrombin from the antibody. So basically, this generates on and off rates, and we can see the actual binding affinity. So when we look at the binding affinities here, we can see here in yellow is the wild-type binding affinity. We have 2 mutants that we could not accurately assess the binding affinity, just simply because the binding was too low, which either could indicate that these mutants are in a much more closed conformation, but it also means that the mutation disrupted the binding site. So that's something we are currently still investigating. Now in this graph, the lower bars actually mean a better binding. I'll finish up after this. Yeah. And we can see that 4 of the mutants displayed a better binding affinity, meaning that these are actually shifted to a more open conformation. So just like to finish with this take-home message. Protein biology is a bit more complicated than we thought. I hope I could show you a little bit that proteins don't exist in just identical copies, but actually in a heterogeneous mix of different structural forms. And with that, I'd like to thank everyone involved in this study and our collaborators. Thank you for your attention. Thank you.
Thank you for that presentation, Mark. So next up, we've got one of the last of our in-person speakers, and that is Dr. Amy Jiang, who will be talking about her hybrid mu and PVA-MA hydrogels for vascular grafts.
Thanks for the intro and thanks for having me here today. So today I'm just going to talk about some of our latest work on the vascular graft biomaterials. So I'll just get started. So atherosclerosis is the major cause of vascular diseases, accounting for roughly 30% of the death worldwide. And unfortunately, this number is still increasing due to the aging population and also the current lifestyle. So this poses a large healthcare burden that roughly 800,000 vascular bypass surgeries are carried out each year. So currently there's two graft material options. One is the autologous blood vessel. So that's an ideal case. That's a segment of the blood vessel from the patient's own body. But however, for patients who do not have this vessel available due to age or pre-existing medical conditions, they have to go for a synthetic approach. So they have to use materials such as Garvan or EPTFE. As you can see, these are the same materials that we use for the plastic drinking bottles and the non-stick Teflon cooking pan, so not ideal. And this accounts for roughly 40% of the patient cohort. These materials are not too bad when they're placed in a large diameter blood vessel. However, they uniformly fail when they're used in a small diameter blood vessel, meaning the inner diameter is less than 6 millimeters. And this technology hasn't really been changed for the last 50 years. So there's an urgent need to develop a new artificial vascular graft that blends the cost effectiveness of the synthetic materials as well with the biocompatibility of the autologous blood vessels. So there's a few emerging techniques. For example, the electrospinning. It's promising because it produces nanofiber structures that mimics the extracellular matrix. However, they tend to have a smaller pore size that limits the cell infiltration and integration. The other approach is by casting or bioprinting of hydrogels. Hydrogel is good because they provide a biomimetic environment for the cells. However, they tend to have low mechanical strength, so that's why they can't be used as a standalone material. Therefore, to overcome these limitations, we've designed a hybrid scaffold. So a little while ago, Professor Thomas Youngster from the University of Würzburg, Germany, who is an expert in mu, met with Professor Kun Lim, who is a leading researcher in the PVA-MA hydrogel space. And when two researchers meet, of course, they come up with a great design. Why don't we combine the best of us? So this is promising because the mu provides a highly organized architecture and also a defined porosity and geometry. So it strengthens the mechanical side of things. Whereas the PVA-MA-based hydrogels, they are photo cross-linkable, therefore provides a precise spatial and temporal control, and also has a tunable mechanical properties as well as high batch-to-batch reproducibility. So the combined formula would have a structured, biocompatible, and mechanically tunable vascular graft biomaterial. However, PVA-MA is quite bioinert. To overcome this, we've started blending in biological polymers. So we added a little bit of heparin, so that's the methylated heparin, in hope of reducing material thrombogenicity. And we also added some gelma in the hope of enhancing cell interactions. And if both of these hypotheses holds true, then the combined material would be promising for the vascular graft applications. So Verena, who was a summer exchange student, came to our group and did some of this work. So looking at the first hypothesis, which is looking at the blood interactions, we first did a whole blood adhesion assay where we incubated samples with heparinized whole blood for up to an hour under shaking. And the samples were then washed in PBS, imaged to look at the fibrin coverage, and also was quantified as shown on the right. So here stainless steel was used as a pointer is now. There. So stainless steel is used as a control and is quite thrombogenic, as you can see that as early as 15 minutes, it's got quite a bit of fibrin coverage and progressed to a full fibrin coverage by 60 minutes. P10, so which is this material, so that's the PVMA alone, isn't too bad to start with, but so slowly progressed to a lot of fibrin deposition by 60 minutes. The addition of heparin completely eliminated the fibrin formation on these scaffolds. But in contrast, the addition of JAMA made it made it a little bit worse compared to the PVMA alone. However, but with the help of a little bit of HEPMA again, it was able to bring down this fibrin formation quite significantly, as shown on here as well. From this, we can tell that the HEPMA reduced fibrin formation on these P10 Barrett scaffolds over time when it's in contact with whole blood. However, this experiment was done under shaking, so it's quite low shear. So we want to see how it performs under flow because that's more physiologically relevant. So to test this, we did a modified channel loop assay, so where we assembled the samples in the loop and then injected heparinized whole blood into the loop and then incubated on rotation for 30 minutes. After that, the samples were washed and the clots were imaged. So this result is quite consistent compared to the whole blood adhesion as well, so that the stainless steel was quite thrombogenic. It has a lot of fibrin or clot formation on the sample material as well as in the flowing blood. Whereas the gelatin was also quite thrombogenic as well. So you can see there's a lot of clot formation on the sample as well as a clot fished out from the flowing blood as well. However, for the samples in the presence of HEPMA, they have rendered these constructs clot-free. We did NQ3 and we did 4 different donors to account for donor variability. And we can see that the samples in the presence of HEPMA have consistently showed the lowest amount of clot formation. So with this, we can conclude that the HEPMA reduced clot formation on these scaffolds under flow as well. Therefore, we get a tick with the first hypothesis. Then moving on to the second hypothesis, which is looking at the cell interactions. We first did a cell adhesion assay, where we incubated endothelial cells on the TCP control as well as the various scaffolds. So control, as you would expect, there's a lot of cell attachment. P10, there's very little cells attached because it's quite bioinert. HEPMA didn't really change much, but the samples with the presence of JAMA have shown a significant increase in the cell number of cells attached. So this we can say that the JAMA did promote endothelial cell adhesion on these scaffolds. We also looked at endothelial cell proliferation as well. So again, we seeded the cells on these scaffolds and cultured for 3 days as well as 6 days. And interestingly, that both the JAMA and HEPMA have enhanced this endothelial cell proliferation. So therefore, we can conclude that the JAMA and HEPMA promoted endothelial cell proliferation, and therefore we get a second tick with the second hypothesis. So with that looking promising, we want to see how translatable is this in 3D because that's more clinically relevant as well. So we then had these tubular scaffolds and then we seeded endothelial cells on the luminal region and incubated for 3 days. And then we imaged, we stained the cells with active filaments in red and then cell nuclei in blue. And we imaged from the both the luminal region as well as, so the, oops, sorry. So we looked at both the luminal region as well as the cross-sectional area, and we can confirm that a successful endothelium was formed on these 3D Barrett scaffolds. So therefore, we can conclude that the PVMA, with the help of HEPMA, we can reduce material thrombogenicity. With the help of JAMA, we can enhance endothelialization. Therefore, the combined material is promising to be used as a vascular graft application. So with that, I'd like to thank my supervisor, Anna Waterhouse, as well as the collaborators from Kuhn's group, as well as Thomas's group, and also the funding bodies as well. Thank you.
Thank you for that presentation. I will introduce the next speaker, which is Dr. Nicholas Hindley, a Cancer Institute New South Wales Early Career Fellow and an Honorary Fellow of the National Heart Foundation, Nicholas is currently leading an international research program driving machine learning advances for image-guided radiation therapy. And by doing so, Nicholas hopes to make cancer treatments available to everyone at a very low cost. And to begin towards that goal, has developed 2 open source algorithms for real-time cancer targeting. Okay, yep, the floor is yours, Nicholas. Thank you for being patient.
Cool. No worries. Thank you for handling the tech issues. And, you know, a special hello to everybody at the Garvan and the Victor Chang, because I grew up in that area. So when I was a kid growing up, I always saw the Victor Chang and the Garvan as these magical places where discoveries happen. So it's quite nice that I get to be addressing that audience. But I'm a radiotherapy guy, as was mentioned, and this is a cardiovascular audience. So you might be thinking, why am I speaking to you? And I think this paper, this paper out of the New England Journal that was published in 2017 is a really nice encapsulation of one of the possible relationships that radiation therapy has to cardiovascular disease. So you can see on the y-axis here, and so we have data for 5 patients, and you can see on the y-axis the number of ventricular tachycardia episodes. So these are patients with these terrible arrhythmias, and I suppose that the frontline treatment are antiarrhythmic drugs. And if those don't work, we will escalate to the current gold standard treatment, which is catheter ablation. Unfortunately, that procedure is sort of invasive and costly, and also has sort of lower success rates than we'd like and probably higher complication rates than we'd like. And so these are patients for whom all of the available treatments have failed them. And so what we've done is we've delivered a single fraction of radiation therapy. So, you know, with a typical cancer patient, there'll be multiple rounds of radiation therapy. And in this case, we only deliver a single treatment session. And what you can see, and the x-axis here is months after treatment, what you can see is that after that treatment, there's about a 2-month period where we sort of reduce the number of ventricular tachycardia events. And what we think is happening there is that the cardiac tissue remodels itself, you know, in response to this insult of radiotherapy. But once that's happened, essentially these patients show 0 number of cardiac events. And I'm told that, you know, with these patients, you know, quality of life is absolutely terrible when you don't know when this ICD thing is going to go off. And here we deliver a single fraction of radiation therapy. They sort of come in the morning, they get imaged, they get treated, and then they can leave in the afternoon. It's essentially outpatient and incredibly inexpensive because it sort of leverages everything that we already have in place for radiotherapy treatments. So that's one of the possible relationships that radiation therapy has to cardiac disease. More commonly, though, the heart is not a target. In fact, the heart is the opposite of a target. It's a critical organ at risk. We know that cardiac tissue is particularly radiosensitive. And so this is of particular concern for lesions that are close to the heart, such as breast cancer and lung cancer. And essentially what we know is that if you have an increased collateral dose to cardiac tissue, there's a higher likelihood of cardiac disease. And so even in the case where the heart is a target, really you want to be irradiating a very, very small subset of the heart, a very particular part of the heart. So for instance, if you're thinking about atrial fibrillation, and you know, I've got a cardiac audience, so I can speak technically, the target there is the pulmonary vein antrum. So it's a very, very small subsection of the left atrium. And then if you've got ventricular tachycardia, you're probably looking at scar tissue around the ventricles. So even if you are trying to hit the heart, you're trying to hit a very small subsection of the heart. And then when you're not trying to hit the heart, of course, you're trying to avoid it entirely. And so that's where image-guided radiotherapy really comes into play. And what you can see at the top here are these images that are being acquired for a patient while they're being treated with radiation therapy. And so you can see at the bottom, like in that acquired projection at the bottom, you can see the diaphragm and you can see the heart beating and you can see that respiratory motion. And so these cardiac targets are arguably the most sort of complicated, most difficult to treat targets in all of radiation therapy because you've got this combination of respiratory motion and cardiac motion. And so I've developed this deep learning framework called voxel map that takes those 2-dimensional images that we acquire during treatment, and it predicts the underlying 3-dimensional reality. So here we're confronted with what in mathematics is known as an ill-posed problem. And that is one where we have way more unknown variables than knowns. So we know the 2D images, and what we would like to know is the 3D motion. So as I say, we've got a sort of higher dimensional solution space than we do our input space. But this problem, and I'll show you how this works in just a moment, is now being solved with deep learning. And you can see that we're estimating the underlying volumetric images at the bottom. But we're also estimating the sort of the position of the target, which in this case is the left atrium, and as I mentioned before, a very specific subsection of the left atrium. And we're also estimating the position of the surrounding organs at risk. So in this case, those would be the lung, the stomach, the spinal cord, and the esophagus. Now, I promised to say how this works, and it was very important to me that whatever technology we come up with, it can be seamlessly integrated into the existing clinical workflows. There are sort of competing systems that, they basically require dedicated and expensive systems that are not available in 95+% of treatment centers around the world. And so it's very important to me, like an additional constraint was to say, well, this also has to work on a standard radiotherapy system. And what will typically happen for a patient that's being treated for a lesion, and this would also include cardiac radioablation, where we know that the target is experiencing significant respiratory motion, will acquire what's called a 4-dimensional CT. And so that's like your typical CT, but if you just try and take a regular CT for a patient while they're breathing, the image will be blurred. For the same reason that if you try and take a picture within a moving car, the image will be blurred. And so what we do is we acquire we acquire a normal CT, but we reconstruct volumes using slices that were acquired at the same point in the respiratory cycle. So you can imagine, okay, every slice that was acquired when the patient was at peak inhale, we take those, we reconstruct a volume. Every slice that was acquired when the patient was at peak exhale, we reconstruct those as another volume. 20% inhale, 20% exhale, et cetera, et cetera. So instead of having one very blurry image, you have 10 what are called respiratory correlated, less blurry images. And my basic proposal is that there's actually a lot of information in those scans that we're not using to their full extent currently. So first of all, what we can do is we can track all these structures over that particular respiratory cycle using a technique called deformable image registration. And that gives us an idea of internal 3D patient motion. But in addition to that, once we have the 3D volumes, we can then ask questions like, well, if the patient's at peak inhale and we image them at 30 degrees, what are they likely to look like at 2 dimensions? And so the picture that I'm trying to paint is that from this 4D scan, we then get basically a perfect training data set of 2-dimensional images and 3-dimensional motion, which can then be used to train a neural network to understand the relationship between the two. And I should say this is done in a patient-specific manner because we're essentially pattern recognizing when the diaphragm moves this much in 2 dimensions, how much does the lung move in 3 dimensions, etc. And all of those relationships depend on the particular biomechanics and anatomy of the patient. So once that's trained, we then, on the treatment day, we acquire these images on the radiotherapy machine, we pass them through that trained neural network, and then we can predict 3D patient anatomy, as you saw earlier. Now, from the perspective of a neural network, an image is just a matrix. It's basically just a spreadsheet of numbers. And so I've sort of shown you that this works in the X-ray case, but actually, yeah, there's no reason that this doesn't work on MRI. In fact, it works better on MRI because you get this improved soft tissue contrast. And for those of you that are MRI experts, you'll also know that when we acquire MRI, what we're actually doing is we're collecting points in what's called K space or frequency space. And it turns out that with VoxelMap, you can skip image reconstruction entirely and go straight from the raw data to 3D motion. So we've now had a couple of publications in this area, and what we've been able to show in silico is you can achieve a dice similarity to the target. And if you don't know that metric, what I'm basically saying is percentage overlap. So we get 80% overlap between our predicted and ground truth targets. And that goes all the way up to 90% for the surrounding organs at risk in the MRI case. And we can also predict the underlying volumetric images or 3-dimensional images with a structural similarity to the ground truth of about 90%. I mentioned lung cancer as a particular site where these cardiac events are a particular issue. And what we've shown in patient data is that you can achieve a tumor localization error of sub-millimeter accuracy. And also you get sub-millimeter accuracy for the surrounding organs at risk as well. And so to sum up, what we have here is a method for real-time 3-dimensional motion estimation and volumetric imaging that, as I mentioned, is very flexible. It works just as easily on MRI as it does on X-ray. And as I mentioned, we've now published our results showing that this works in silico and retrospectively on patient data. But the two questions are, can this be implemented prospectively on patients? And, does this work on a clinical system? And so I'm now working very closely with the clinical teams at Royal North Shore Hospital and also RPA. And then lastly, but perhaps most importantly, if anybody's interested in using these techniques, all of the results that you saw were generated using code that is freely available at our GitHub. And there's a QR code there if you'd like to check out the code. And in addition to that, if you don't have a background in machine learning, I've put together a tutorial that takes you line by line through how the code works. So with that, I will hand back over to the people in person and looking forward to questions.
Thanks, Nicholas. So next, I'm going to introduce Dr. Eden Barrett, if you would like to start sharing your slides. Eden is a research fellow and dietitian in the Food Policy Division at the George Institute for Global Health and a lecturer at UNSW. Her research aims to inform public health policies, with one recent example of the research being a Heart Foundation Vanguard grant project she led called Grain Switch, targeting online grocery shopping to encourage heart-healthy grain choices. If you'd like to unmute your microphone, Eden, and take it away.
Thanks, Jordan. The audio is okay?
Yep, all good.
Great. Thanks for joining today, and I'm excited to talk about this work. It's a bit different to some of the work we've seen so far, so I hope it's still interesting for everyone. We're talking about food. So to start my talk, I would like to ask you to all imagine you're in the grocery store or maybe the online grocery store and you see these three breads. From looking at them, would you know which one was the highest in whole grain? And if I was in person, I could get a show of hands, but I'm not. So I'll just ask you to think about it yourself and it becomes relevant later. So the reason I care about this is that I think low whole grain intake is probably one of the more underrated components of a healthy diet. We know that low whole grain intake is actually one of the leading causes of diet-related death and disability. It's in the top 2 with diets high in sodium. And studies show that 3 serves a day lowers risk of heart disease by about 25%. And can actually reduce blood pressure and cholesterol, similar to what is achieved with drug therapy. So super important, but people don't really know it. They know kind of eat more fruits and vegetables, eat less salt, but people seem to forget about whole grains. And we know that because people aren't eating enough of them. So in Australia, people eat not enough whole grain and they eat too much refined grain. So refined grains are white bread, white pasta, white rice. Up until a week ago, this was the most recent Australian Health Survey data, and it showed that we were eating about half on average the recommended intake of whole grains. Survey did come out last week rather, so it'll be interesting to see what that shows. But I suspect it will still be lower than recommended. And there's a few reasons why people don't eat enough whole grain, we think. One of them is taste and texture. People prefer the taste and texture of white grain foods. But there are other things at play as well. And something that we think contributes is that it's actually quite hard to find good sources of whole grain foods in the supermarket. And contributing to that is the fact that in Australia, the labelling is not very good. It's not very regulated around whole grains. So FSANZ basically says that as long as the product contains a whole grain ingredient, then it can be labelled and called whole grain, right? Whole grain on the packaging. And the result of this is that you can have products that are seemingly a whole grain bread, and one might have 35 grams of whole grain per slice, and one might have 2 grams. So given this problem, we wanted to test the impact of a whole grain label that showed whole grain content per 100 grams, as well as a color-coded indication of really high whole grain choices. So best choice is what we called them in green. Better choices, yellow, and then if a product was low in whole grain, red. And we tested this in an online shopping choice experiment. We had 2,000 Australians and we randomized them into 2 groups. And both groups in the study saw 3 different products across 4 food categories. So the food categories were breads, breakfast cereals, savory snacks, and rice cakes. And so the intervention group saw the whole grain label and the color code label, and the control group didn't see that information at all. They just saw the products. And then we asked participants to select the product that they would choose and to also rank them on healthfulness from most to least healthy. So this is what that label looked like in the online setting, roughly. So you can see these are the breads from before. Is that the order that you thought they were in? And also, is it the whole grain content that you thought they were going to be? So that's what the intervention group saw. And the control group just simply saw the breads and their name. So like I said, we had just over 2,000 participants complete the experiment. Generally, they did kind of look similar to the Australian population. The only difference, or the main difference, was they're a bit more educated. So we had 45% of participants with a university degree. We know that is more than the average Australian population. But the good thing is the characteristics didn't differ between the control and the intervention group. So we found that up to 80% more people chose the best choice option when the labels were provided. And this differed a little bit across the 4 categories that we looked at, but all of the differences were significant in the intervention group compared to the control. And similarly, up to twice as many people correctly ranked the product healthfulness. Again, it differed a little bit over the food categories, but all were significant. We did test a few things as potential moderators in the relationship. So we tested age, gender, education level, and reported health conditions. But none of them showed consistent moderating effects, which means that labeling appeared similarly effective across diverse groups of people. Something that we did find, which was kind of shown on the earlier slide, is that there did seem to be some variation in findings between food categories. And we found that the effects of labeling were weakest for the savory snack group. And we thought about why this might be. And firstly, as you can see what these products were up the side, there's probably a bit more diversity in them than, say, between breads. Maybe it's easier for people to choose a different bread than it is to go from something like a potato chips to popcorn. And the other thing that we think perhaps as well for these type of products, seen more as an occasional food, health information in general might be lower priority for people than maybe when they're picking breads and breakfast cereals. So in conclusion, we found that color-coded whole grain labels and whole grain content information positively influence choice and healthfulness perceptions. Obviously, this was a very kind of crude hypothetical experiment online. The next step really is to test this impact in real-world shopping settings. I didn't say it earlier, but we didn't show things like price, and we also tried to keep the brand as much as possible consistent within the choices to avoid the impact of those factors as well. But we know that in real life, they're going to impact choices. So this on the side is kind of our ideal tool that we're looking to develop, where when people are shopping in an online setting and they pick a product for their cart, they are provided suggested switches for higher whole grain content. And then the last conclusion, I guess, that come from this research is that because people do take note of these labels and they do trust them, it's really important that products that are promoted as high as whole grain through labeling must be genuinely high in whole grain and meet minimum nutrition standards. So this comes back to the regulation not being particularly strong in Australia and helping people to make good choices with labeling that they see. And the other thing that we did as well is we made sure that the best choices that we suggested, they all had a health star rating of at least 3.5 and also had a health star rating that was higher than the other products as well. Because again, obviously whole grain is one really important element, but if we are going to promote foods, we also want to make sure that they're across the board meeting minimum healthiness criteria. So that is all from me. I'd just like to say thank you to the wonderful team that contributed to this work. And on that team, we also had 2 really great consumer representatives that helped us put together the survey and interpret the findings. Thanks.
Thank you, Eden. We're now going to switch back to in-room presentations. All right. The next speaker is Fairooj Rashid, a postdoctoral researcher at the Westmead Institute for Medical Research. She's recently completed her PhD in the immune stromal cardiac interactions, focusing on their spatial presence using a new technique called imaging mass cytometry. So I'll hand it over and we'll learn more about it.
Thanks for the introduction. Today I'm going to talk about my PhD project that delves into spatial biology of end-stage heart failure patient samples. So heart failure isn't just an issue with the pump, but it's a progressive disorder affecting over 64 million people globally. Despite advancements in therapeutics, prognosis remains poor for many, and this is due to our lack of understanding of how various cardiac cells communicate and evolve with disease progression. So my research uses spatially resolved single-cell proteomics to decipher these interactions to better understand heart failure progression. So single-cell transcriptomics revealed diverse cellular subtypes in end-stage heart failure patients in normal heart samples, starting from endothelial, myeloid, lymphoids, fibroblasts, but they do lack the localization information. Hence, technologies like imaging mass cytometry are essential. So IMC allows single-cell multiplex protein detection, retaining spatial resolution, allowing us to connect cell identity with the tissue architecture. So to better understand heart failure microenvironment, my project was stratified into 3 core aims. The first was to build a robust cardiac-specific IMC workflow optimized for fibrotic heart failure myocardial tissue. The second was to delve more into the biology, dissect cellular phenotypes across the fibrotic microenvironments in ischemic and dilated cardiomyopathy. And the third aim was more for the bridge to translation to identify new spatial and molecular signatures informing future diagnostics or therapies. In this study, we collected left ventricular apical cores from 8 ischemic and 8 dilated cardiomyopathy patients undergoing LVAD implantations. A blinded histopathologist scored the different regions within those myocardial samples in according to fibrosis, starting from interstitial to replacement to compact. And then within those regions, I selected areas with intent on heavy cellular activities for downstream analysis. Using a 37 marker panel tailored to heart failure, we acquired 3,400 multiplex images with 1 micron resolution. And using this high dimensional data set, we applied unsupervised clustering to look into the cellular phenotypes and single-cell spatial analysis for around approximately 4 million cells across 185 millimeter square area of patient tissue. To optimize an IMC's heart failure specific IMC workflow, it comprised of 2 important parts. The first is cell segmentation, which is of vital importance when it comes to spatial biology. Now, cardiac tissues present with unique challenges due to the different types of cells in its microenvironment. To address this, I developed a machine learning improvised cell segmentation pipeline by training the algorithm on the various cell membrane features. And for the first time, we were able to segment all the various cardiac cell types. And for every individual cell, we acquired localization information and also panel marker expression. The second challenge was to assemble a 37 marker panel which can capture heart failure biology. So the panel consisted of metabolic, immune, vasculature proteins, and all these proteins were carefully selected, validated, and optimized for this study. Something that was really interesting was to see how cellular fibrosis in these end-stage heart failure patient samples were. Like clinically, we would expect scar to be acellular, but upon applying this single-cell spatial technology, I actually saw that scar teams with various immune and fibroblast cell subtypes. With the high-dimensional data set, we were able to profile stromal, myeloid, and lymphoid cell types. But for this talk, I'm just going to talk about the cardiomyocyte subtypes that emerged from our analysis. It was quite interesting to see that there are two subtypes of apoptotic cardiomyocytes. C2 was predominant in DCM patients, whereas C3 was predominant in ICM patients. And it's quite evident from this bar graph showing individual patient cardiomyocyte subtype abundance data. One of the biggest observation from this analysis, which later we reckon it's a discovery, was the emergence of C4 cardiomyocyte phenotype. So this phenotype of cardiomyocytes, along with sarcomeric protein expression, they were also expressing a glycoprotein, which I've termed as marker X. Due to proprietary reasoning, I'm unable to reveal the name of this protein. But when we looked in the literature, it was never reported that cardiomyocytes can actually express this protein. And I'm going to address this phenotype later in the talk. So I've just shown that how cellular scar is, and this led to the question, is scar or fibrosis in end-stage heart failure patients structured? And the answer is yes. So when we sort of spatially mapped all the cell types back into the tissue, we saw the emergence of specific cellular niche or neighborhoods. I would like to draw your attention to niche 4. And this was quite interesting because niche 4 did not only comprise of all different types of pro-inflammatory cells like cytotoxic T cells, B cells, neutrophils, but at the same time, they're only predominantly present in replacement fibrosis, but not in interstitial or compact fibrosis. And this actually shows that this can be a potential therapeutic target. With all the different 4 million cells we have profiled, we were able to perform single cell-to-cell interaction. And you can imagine the amount of data that we acquired. So this ball plot is just an overview showing the changes in cell-to-cell spatial interaction when we compare ICM with DCM patients. Red shows attraction, whereas blue shows avoidance. Just to give you context, this is an example. So the C4 novel cardiomyocyte phenotype that I mentioned previously, it was seen that this cardiomyocyte subtype were closely associating themselves with fibroblast population 3, which is a fibroblast population that was expressing fibroblast activated protein, and they're quite lethal in small animal models of MI. Now, back to the C4 phenotype. So we observed it in IMC. Then the next question was, what is protein X? So it's a glycoprotein expressed by lymphatic endothelial cells and also some macrophages. They were involved in cell motility and cell autophagy. So it was quite bizarre to see that cardiomyocytes were capable of, some cardiomyocytes, not all, were capable of expressing this protein on their membrane. So we performed a high-resolution confocal microscopy and then stained the tissues with other endothelial cell markers like, let's say, CD31. And as you can see, the cardiomyocyte in red, some of them are actually expressing protein X on their membrane, whereas they're not expressing CD31, which is an endothelial cell marker. And that kind of was providing us a validation it's actually a phenotype. And then we looked into mining data from published data sets to see if this phenotype is also abundant in publicly available data. We mined, as I mentioned, we looked into 1,200 myocardial heart samples from donor and ICM patient samples, and we did find that, yes, this phenotype actually exists in those data sets. Now, the discovery of this C4 phenotype expressing protein X warranted further biological functionality exploration. So we went on using AAV6 to manipulate the protein X expression in iPSC-derived cardiomyocytes in vitro. So when we transduced these cardiomyocytes at day 25, we saw that the group, which was overexpressing protein X, had increased cell proliferation, as evident in the images. So red is Aura B kinase. It's a protein that gets expressed during cell cytokinesis, like i.e. cell division. And it was quite interesting that these cell groups, they were more proliferative in nature as opposed to the other groups. In the same set of experiments, we put the iPSC-derived cardiomyocytes day 25 under ischemia reperfusion injury model. And then we saw that the group, which was overexpressing protein X, had better cardioprotective functionality. So it was very interesting to see that, protein X was kind of protecting them against hypoxia. So this leads to more in vitro validation experiments that we need to perform, maybe some small animal work, but this is quite interesting. And to actually have everything starting from end-stage heart failure patient samples coming up with a finding, and then to look into in vitro cells, it was quite interesting, and I think it was a pretty good experience for my PhD as well. So to summarize, so this project shows the first patient-resolved atlas of human heart failure at single-cell resolution. We did see some differences in the heart failure microenvironment that is disease etiology-driven in ICM and DCM patients. And we were able to characterize some distinct fibroimmune niche. And then there was the discovery of the C4 phenotype, which actually showed that in vitro, they do retain some proliferative and cardioprotective functionality. I want to thank my supervisor, Professor James Chang, and all our collaborators across Australia for helping me with this project. Thank you very much. Thanks.
Yeah, thank you for that really good talk. Next up, we have Merce Fuentes Amell, a PhD student in human physiology at the University of Newcastle. Merce focused on cerebral spinal fluid and the dynamics under elevated intracranial pressure. But I'm sure you'll be able to there's a little bit more than I can do. I will. Is this one yours? There you go.
Okay. Hello, everyone. Good afternoon. My name is Merce, and I'm going to be talking about the impact of hypothermia in stroke, and I'm going to be presenting a proteomics study. So to start with, we have that hypothermia is one of the most effective strategies in preclinical studies. When we talk about hypothermia, we're talking about like 35 degrees and less. And it's very effective in reducing infarct size, and it also protects against reperfusion injury. And some groups have been also focusing in short duration hypothermia and have found some effects as well, which we will discuss later. In humans, though, there was a very important study named EuroHype study that tried this treatment, but however, they could not achieve the target temperatures, and so it wasn't effective. However, when we focus on what we know about hypothermia, we can see that these are the known effects, which are like reduced cerebral metabolic demand, reduced oxygen consumption, and like these effects that we can see in the image, which are kind of like long-term effects. But like I said earlier, some of the groups were looking at short duration hypothermia, and in those groups there was our lab, and we found that short duration mild hypothermia had long-lasting effects. And I couldn't put it here, but they were like switch-like. And this meant basically that this short-duration hypothermia is triggering some changes that haven't been characterized yet. And so that's why we designed a proteomic study in order to identify these changes and find new potential therapeutic targets that can be applied in humans. And so for this study, we designed 2 treatment groups, which would be sham or stroke in rats. And then we either treat them with hypothermia or normothermia. And then we investigated each of the hemispheres, so the stroke or ipsilateral hemisphere and contralateral hemisphere separately. As for the methods, we collected the brains and cerebrospinal fluids of these rats, and we used the brains to quantify the stroke volume, but we flash froze one slice for proteomics. And in that slice, we took samples from the ipsilateral penumbra of the stroke and the matching contralateral side. And then we digested and separated those proteins, and we ran them through mass spec, label-free mass spectrometry. And we use this device called FAMES, which basically uses compensation voltages in order to gain sensitivity and more precision in our samples. And for this seminar, I'm just going to be presenting the -60 voltage results. And so after that, we ran some bioinformatics, and we're going to talk about like the exciting part, which are the results. And so the first thing that we found is that hypothermia reduced edema-corrected stroke volume compared to normothermia. We also found that the stroke-induced proteomic changes affected both hemispheres, and therefore the contralateral hemisphere is not an appropriate control for molecular studies, which is the current standard in the field. We also found out that hypothermia activates regulatory pathways involved in cell cycle, cell death, and degradation inflammation in the ipsilateral hemisphere, and that in the contralateral hemisphere, hypothermia attenuates the stroke-induced molecular changes and also the post-translational modification in proteins. And finally, for this project, we want to analyze across the cerebrospinal fluid and all of these voltages, and in order to identify, like I said, the target molecules, and then use in silico and in vitro techniques to find drug candidates to test those targets. And finally, I'd like to thank my supervisors, Kirsten and Niels Pratt, and everyone else in the stroke lab and the University of Newcastle. Thank you.
Thank you. And now we have our final speaker, Cristina Zeng, a registered nurse and a PhD candidate at the School of Nurse and Midwifery at UTS. Her research focuses on cardiovascular disease prevention, management, particularly among cultural and linguistic diverse groups. I'll pass over to Cristina.
We just now talk about a lot story in the lab and also in the supermarket. Now I will talk something about people like in the community. Hello everyone, I'm Cristina. I just finished my PhD in nursing at UTS, and today I will present part of my PhD result. In hospital, I always came across some Chinese patients. They are quite struggling with two different cultures and healthcare systems. So they result in poor outcomes. And it has been widely reported that this group has poor CVD profile compared to the host population and people in China. And also little is known about their self-care behaviors, which is the cornerstone for CVD secondary prevention and management. So all this brought me to think, how do Chinese immigrants, they look after their CVD in this cross-cultural context? Sorry for that. Yeah, it is important to address these questions as it can improve healthcare providers' culturally competent practices and inform the development of CVD self-care interventions. We use exploratory sequential design, and I recruited 280 participants from the community settings. During the data connections, I also give the heart health talk because I want to give something back to the community while I collect data from them. So this is part of my phase 1 and phase 2 result. They are consistently indicated that although Chinese immigrants, they actively engage with their self-care behaviors, predominantly in self-care monitoring, self-care maintenance, but many of them are not evidence-based and also inadequate, particularly in response to heart attack and stroke. So, and our qualitative and quantitative findings the findings both say that, sorry for that. Also, the qualitative and quantitative findings, they both explain the factors that impact their self-care behaviors. So notably, and confidence in CVD health knowledge and searching for Western source health information was significantly associated with better self-care. But less than 1/3 of them reported feeling confident in their CVD CVD knowledge and access to Australian health information. So this all imply an urgent need to like develop and deliver linguistically and culturally appropriate health education resources to this population. And collaboration with the community resources such as Chinese community associations would offer innovative approach to co-design and delivery this kind of CVD health information. Thank you so much for that.
Thank you. So that concludes the presentations for today. We can now have a little bit of time for questions, but I have been informed that the ceiling microphone is not picking up audio. So potentially, if you've got a question, it might be best if you come up to the microphone at the front so people online can hear the question as well. Does anyone have any questions to get us started? Well, I had one. That was actually for you, Albert, with your presentation. I missed a little bit of it due to some technical issues. In your discovery, you discovered quite a lot of new sort of novel hits. And, you know, this is definitely a topic where other researchers have been investigating this throughout their careers. Could you comment sort of what was it about your approach that let you uncover, I believe it was 30 odd novel transcripts? Yeah, why do you come? Sorry, might be easier.
Yeah, so just to confirm, the finding then, the discovery is about genetic variants. So we're essentially analyzing through a genome-wide association study, collecting cases from different cohorts, and then we perform genetic association analysis and identify those. In the end, we link genetic variants to the gene. So it appears that there are like novel genes, but those genes are actually identified through variations. Oftentimes in the nearby of the gene regions. But in terms of why it identifies sort of like incrementally higher number compared to previous studies, I think it's a combination of like multiple things, but we believe one of the biggest reasons, because we did a similar study in 2020 with about like a third of the case that we have, and we identified fewer genetic loci. So one obvious factors there is a simple increase in the number of cases we identified. And that was through collaboration. So we didn't collect the data ourselves. We collected data from multiple cohorts globally, and we did some sort of like meta-analysis. So I think that's one big change. The other things I would say, it also depends on the phenotype definitions, because we're looking at the heart failure, which is a very heterogeneous phenotype. And we found there's a trade-off. If you go very broad definitions, you increase the number of sample, but also there's a chance that there are lots of mislabeling or misclassification. So the ideal way is to maximize the sample size, but also be as precise as possible with the phenotype definitions. I think that would probably give kind of like the highest number of discovery. I hope that answered the question.
Yeah, yeah, absolutely. I guess managing to get that huge data set through multiple collaborators. Yeah. And then I suppose you're relying on more and more different clinicians to accurately label their data set. Yeah. So
which is a challenge, because especially if you go to like multiple in different countries, sometimes the definition, it has like slight difference. Even the same countries, different hospitals, sometimes different clinicians have their own way to define. So that's the challenge.
Yeah, absolutely. Do we have anyone online, Mari? So we've got a question for Amy. So perhaps I should probably turn up the volume on the computer so you'll be able to hear the response as well. Would like to know, what are her next plans for validating the hybrid vascular grafts? Are there any in vivo models of vascular disease that you're considering?
Thanks for the question. So so right now we just tested the material itself. We tested the acute response from the blood and the cells. So that's very early stage. And the next step will be looking at the hybrid scaffolds, how like the mechanical side of things, so that we can tune the mechanical properties similar to the native vessel. And also in terms of animal model, we do have a couple animal models we've done before. So one of the models we've done was a mouse carotid artery interposition model. So we basically you suture that into the carotid artery and implant for like you can implant for up to 30 days to look at the endothelial dilation and also intimal hypoplasia, that kind of side of things. So that's a small model, and there's also large animal models like the sheep. So we've had those models ready as well. But
Nice.
yeah, right now it's quite early on, so there's plenty to optimize.
Yeah, and just quickly, whilst we've got you on the microphone, I was going to ask you about the endothelial cells that you were showing the proliferation and their ability to bind some of your biopolymers.
Mm-hmm.
Have you confirmed the sort of the phenotype of the endothelial cells? Are they retaining, say, CD31, or are you getting sort of trans differentiation into more stromal cell types?
We have not looked at that yet. So far we're only confirmed adhesion and proliferation. But yeah, that's definitely a next step thing that we will look at.
Yeah, cool.
Thanks.
Thank you. Do we have any questions from the room? Peter's got a question.
I have a question to Mark. That's a really clever essay. So my question is like the carbon 12 and carbon 13 masses. So what can you really like allocate the specific site or like a peptide sequence, like where is the sulfur bond? And also like by adding those reagent, are you breaking the like equilibrium of the like balance of disulfide, that's a formed but disulfide bond or unformed disulfide bond? Thanks.
That's a question here. Yeah, so the first question, yes, we can actually specifically locate the peptides. So what we do in our mass spec approach is we label the proteins with both the carbon 12 and the carbon 13 labeling agents. Then we digest it using any kind of regular enzyme digestion to get like to break up the protein in the peptides.
Short sequence, right?
Yeah, and shorter sequences, because that's the only way we can really mass spec those things. And what we get is like, for instance, so it's breakdown peptides, and then what we see is that the exact same peptide is then one part is labeled with the carbon 12, the other one's part the carbon 13, and we can calculate those ratios with the exact same peptide. That's the real advantage for this technique, because chemically the labeling agents and the peptides are identical to each other. Because the isotopes don't really matter for the chemical composition of the whole peptide and labeling agents. For your second question, no. So the labeling agent can't when there's a bond formed, the labeling agent can't disrupt that bond. So we need to artificially disrupt the bond with DTT. We take that away, and then we can add the labeling agent. So yeah, no, there's no way that the chemistry, that's just not how we know how the chemistry doesn't work like that.
Okay. Okay.
Yeah.
Thank you. Thank you.
Thank you.
We have a question online for Cristina, and that is, what is the most challenging issue when you collect data from the minority group in the community? And do you have any tips for someone that wants to work within that space?
Thank you so much for the question. That's really good question, because when I started my PhD, it crossed all the COVID period. So it was really difficult to collect data from community, especially my population is quite like race and migrate. They just follow their like children's like sponsorship came to the course trainer. So it's really hard to assess this approach. So my skill is like I really work into the community, just doing the start from scratch. I was a volunteer in the Chinese Community Association, so I came across like almost more than 20 like senior groups there. So that's why I had the chance to approach people there. At the beginning, they may not trust you because this kind of like participants, they are quite controverted. They normally don't share their like information or their family things with you. So that's why, yeah, you have to just really go into community. That's why I said during the data connection, I give the hard health talk. I just want to give something back where I collect data from them. So that's my skills. Yeah, sorry. Hopefully it can help.
Yeah, thank you. Do we have any more questions from the room? I think we're running a little bit over time. So if we finish up there, and for those people here in person, we've definitely got a couple of drinks and some nibbles outside. But I'd like to thank all of the presenters for some excellent presentations today, especially the people online for joining as well. Apologies for the technical issues, but we did get there in the end. So hopefully you're online and you're still there just for the moment. Just to catch you, we do have upcoming events. On the 15th of October, it's the EMCR Masterclass. And I believe in the Masterclass, is it the how to write your paper in 3 weeks? So I believe the signups for that will be coming out soon. And I think everyone will agree that's fairly applicable across the board. And just beyond that, in November, if I can remember correctly, we have a more generalized networking event. There'll be a short masterclass style seminar on how to give short, effective communications. And I will definitely be on the lookout for that, which will be circulating through people's inboxes in the next week or two, I think. And with that, I'd just like to say thank you again to everyone, but this time on the actual thank you slide with supposedly a QR code. There you go. Thanks, guys.